So today we will talk about specifically angiogenesis, or the need for the tumor to trick the surrounding tissue into supplying it with a new blood supply in order to keep the cancer cells growing. One thing to think about is that cells in our body, throughout our body, all need to be close to capillaries. You all are aware that capillaries, the thinnest of our blood vessels, run very extensively throughout our tissues, and they're important for supplying gas exchange. So O2 is able to leave the blood through the thin walls of the capillary and get into the tissues. Likewise, waste gases like carbon dioxide can then diffuse in from the tissue into the blood. Also, nutrients can flow out of the blood into tissues, and other metabolic waste products can be collected from the cells and tissues and enter the capillaries to be taken away. So one thing that you may not be aware of is just how extensive your capillary network is. I think when we think about pictures of anatomy, and you see the human body and the circulatory system with all of the main arteries and veins, it seems like it's very extensive, but that's only a small part of it. Because in reality, those types of medical illustrations cannot capture just how crazy extensive your capillary network really is. One thing to realize is that the distance that a cell inside of the tissues of your body can be away from a blood supply is really limited to really how far things like gases and nutrients can diffuse. That might not be something you thought about before, but hopefully it makes sense. Once O2 diffuses out of a capillary, there's nothing really transporting it through your tissues anymore. The gas is just diffusing through all the surrounding cells, and so that distance is very limiting. So typically, something like a gas like O2 will only diffuse about 0.2 millimeters away from the capillary. In other words, that's only about 200 microns. On this slide in our lecture, I've highlighted the micrometer because in the slides that I sent out in the PowerPoint, there was a typo. I think I had put nanometer. But in reality, we're talking about 200 micrometers. That's really tiny. When you think about 0.2 millimeters, that's about as far away as something like oxygen can diffuse out of a capillary. So that means your cells really cannot be that much farther away from the nearest capillary. If you look at this, here is a cross-section of normal liver cells. And you can see these big pink cells are the hepatocytes, or the liver cells. Everywhere where you see a little bit of white space, especially when you see some little red cells in there, those are capillaries. So the little bright red things that you see are the red blood cells that are passing through these capillaries that are sort of cut in cross-section here. You can really see how extensive the capillary network is through there. In places like muscle, right? Muscle cells obviously have a high demand for things like oxygen. Here, these big brown things are individual muscle cells, right? And in between each, these darker stained things are the red blood cells that are passing between them. So of course, these red blood cells are not just floating around between them. Each one of these little white spaces is a capillary that's cut in cross-section. So you can see every muscle cell is nudged up against not just one capillary, but up to 4 capillaries to supply their network, to supply the needed oxygen and nutrients, right? So really extensive network. Not surprisingly, cancer cells, they need a nice blood supply as well, because as the tumor is developing, it has great metabolic need as these tumor cells and this tumor mass is growing. So cancer cells, given a preference, they would love to be close to capillaries as well. Here on the left is an example of a human melanoma that's forming. And so here you have these tumor cells, and you can see they are surrounding a capillary that is providing them nutrients, oxygen, etc. If you move away from that capillary, a radius about 85 microns, what you notice is that the cells that are far away from the capillary are beginning to change. They are beginning to die off and go necrotic because they just cannot get the oxygen that they need. You can look up in the corner here. Here's another example. Here's another capillary, and surrounding it are the actively proliferating cancer cells beyond a certain distance. Beyond that distance where gases can diffuse, the cells become necrotic. Here on the right is an example of prostate cancer taken from a rat. This kind of squiggly thing here is the capillary. So in this particular case, the cross-sectioning of the slide happened to be at an angle where you can actually kind of see the capillary going through. But same concept applies. You go out about 110 microns and the tumor cells are still okay. Beyond that, they're too far away from the capillary. Here's a capillary over here, same thing. Beyond a certain distance, the tumor cells die. They die from necrosis. So this can create a selective pressure then for certain cancer cells to select for random mutations that upregulate things like the Warburg effect. Remember, Warburg effect is when the cells become less dependent upon aerobic respiration because they favor anaerobic pathways like glycolysis, right? So under these situations, you can certainly imagine that the cells out on the borders right here, there would be that pressure for any of those cells to stumble upon Warburg effect type mutations. But the reality is these cells would simply be much happier if they could get a more extensive blood supply to supply them with more blood flow. Here is a good example of what happens taken to the extreme of when you have a very large tumor mass that has gotten way too big for the blood supply to support it. In this particular case, the capillaries would tend to be still in the stroma running by the outside of this tumor, and this tumor is growing. You can actually sense and see the strain, the physical strain that the tumor is placing on the surrounding tissue, where the stroma cells look very compressed and a lot more dense, right? And through that, the capillaries are trying to still flow. So some of these white spots out here in the stroma would be the capillaries. What is happening here is this tumor is getting way too big, so the cells in the middle simply are not getting oxygen anymore. And so you have a massive die-off, massive necrosis taking place in the middle. That's why many large tumor masses are really kind of gross and messy on the inside. They may appear somewhat solid on the outside, but the inside oftentimes is very fluid-filled and filled with necrotic tissue. Right, so in this case, these big white spots in the middle are just empty spaces where the cells have died off, and now it's often just filled with fluid, for example. Okay. When you think about the blood supply coming from capillaries, compared to the blood supply that's already sort of in our large arteries and veins, it's important to realize that our genetic programming, when we are born and our body is sort of laid out in terms of its structure, the layout and positioning of large blood vessels, you know, things like the aorta that comes out of the heart, or the vena cava veins that go back to the heart, the pulmonary arteries, the main arteries and veins that go to our limbs, right? Again, the things that you see on anatomical drawings of the circulatory system, those all come pre-programmed in our genes. Right? So it's not like the positioning of your aorta or your veins and arteries, the large ones, are really all that different than any other human beings. If we think about like, if you have two big cities on a map, here we have in Texas, Dallas, and Fort Worth, the layout of the major highways and freeways, right, is all based upon careful planning. That's all part of the civil engineering, the civil planning that went into allowing these cities to grow and develop and be interconnected. However, if you were to zoom in to this map, what the map doesn't show are all the tiny, tiny little streets and sort of like impromptu little roads that might connect one neighborhood to the next. Those little roads are our capillaries. Right? So those are sort of more ad hoc, meaning they are just kind of improvised and are just kind of designed when needed. So this is kind of the famous Lombard Street in San Francisco, right? The little zigzaggy capillary. So that means major blood vessels are all planned by our genes, whereas all of those capillaries are improvised as needed. So how does a growing tumor then improvise a brand new capillary supply to feed it as it's getting larger and larger? One of the most important signals is VEGF. Right, so we've talked about VEGF before. VEGF is vascular endothelial growth factor. And VEGF is a very important signal in stimulating the growth of new capillaries. Here we have a wonderful electron micrograph image showing the cross-section of a capillary. And what this really highlights is that the capillaries, we talk about them being tiny, we talk about them being thin. You've heard me say before that the walls of a capillary are literally just one endothelial cell thick. And that's what you can see here. Here we see two endothelial cells. The way that this slide is cut in cross-section, you're actually seeing the main nucleus of one cell and part of its cytoplasm that kind of wraps around down here. So this part of the capillary wall, that's all there is to it. It's literally just the cytoplasm of this one cell. This other part over here, you see connecting from this arrow down to this arrow, is part of the cytoplasm of another endothelial cell. And again, just because of by chance the way they cut or cross-sectioned this, you don't see the nucleus of this one. Maybe it's down below or something. And this just happens to be the part of its cytoplasm that was at this particular level where the slide was prepared. But this really shows you, right, that I was not kidding. That's all there is to a capillary, because this space that's in the middle, the capillary lumen, that's where your blood is flowing. And it's very small, a very small confined space where oftentimes cells that are flowing through there, like red blood cells, can only go single file. The space is so small. But really what this shows you is how thin the wall of the capillary is. It is literally just the cytoplasm of one cell thick. Okay. So in order for these endothelial cells to grow and extend a capillary, there are precursor cells or endothelial precursor cells, EPC, that are circulating in your blood, and they will arrive through these capillaries to maybe the end of an existing capillary. And then those EPC cells have to be asked to stop and differentiate and become another endothelial cell to kind of add on to the end of the growing capillary walls. So VEGF then is a very important signal that will allow these capillary networks to form. They also are part of signals that stimulate the growth of lymph vessels. So this is an illustration not taken from our current textbook, but this is one that I use when I teach human physiology and also immunology. But this highlights the fact that our circulatory system, where our blood flows, is not directly connected with our lymphatic system that is in charge of draining various waste products and fluid from our tissues. So in this illustration, the blood is entering from the right side through this arteriole, and this arteriole is dividing into smaller and smaller arterioles until it gets down to being capillaries. And so in this capillary bed over here, you can imagine then that these capillaries right in the middle are just one endothelial cell thick in terms of their walls, right? And it's because of being so thin that gases and nutrients can diffuse out of there. And so that's why we portray it often as the red, highly oxygenated blood. Once it passes through the capillary network and the oxygen has left and diffused out into the surrounding tissues, and the carbon dioxide has diffused in, we then say, oh, the blood is now blue, right, for convenience sake. And then these tiny little capillaries then form venules, and then these venules then join up with other venules to form large veins on the way back to the heart. These green things are the lymphatic vessels. And you can see that these lymphatic vessels are really draped over the capillary bed. At no point in this whole illustration do you actually see a blood capillary connecting directly with a lymphatic capillary. So in other words, the nutrients, the gases that diffuse out of the blood capillary network here and goes into the surrounding tissue, a lot of that is water that leaks out. So the purpose or the function of our lymphatic vessels then is to drain all that excess fluid and waste products from the surrounding tissue and collect it and send the waste material away to be processed. So this is important for the way our immune system works, because it then allows our lymphatic system to collect different types of maybe pathogens or evidence of infection and present it to our immune system somewhere else. But it's also very important for draining things like fluids so that our body can recover all the water that would otherwise build up in our tissues, leading to conditions such as edema, where our tissues get very puffy because of too much fluid. Okay, so this discussion of the lymphatic system is also very important in cancer, because you can imagine different types of cells that might be coming loose in the surrounding tissues can also drain into the lymphatic system as a way of then suddenly being transported elsewhere in the body. Right? So when we talk about metastases of cancer, you can see that that would be very relevant. So capillary and lymph vessel growth is stimulated by VEGF, as well as other growth factors like basic fibroblast growth factor, or otherwise known as FGF. So if you imagine then that a tumor is this crazy, chaotic place where all kinds of signals are going out, including these VEGF signals and other growth factor signals, and it's doing so in a very unorganized sort of way, stimulating the growth of capillaries, it's not surprising then that when a tumor succeeds in recruiting capillary growth, it is very messy and chaotic. So here is a really neat image of the vascular system of a mouse brain in it. And you can see most of the brain is normal vasculature. So stained in yellow are the blood vessels that are supplying the brain. And you can really see how the large arteries that enter branch into smaller arterioles. And then you have a pretty evenly distributed capillary network extensively going through. Up in the upper left corner is where you have a tumor that has managed to learn through mutations how to stimulate angiogenesis, or the growth of capillaries. And you can see, sure enough, how misshapen, disorganized, and chaotic the capillary network is in that tumor. We mentioned that one of the ways that tumor cells trick the surrounding tissue into believing that you need to have the wound healing response activated, and that includes the growth of capillaries, is because the blood vessels in a tumor are very leaky. So on the left here, we have scans of normal capillaries. The green would be the endothelial cells that actually make up the wall of the capillaries. The red are these supporting cells known as pericytes, right? Not parasites, but pericytes. And they wrap around the endothelial cells and help provide some support, some structural support. It's kind of like a garden hose, where if you just had a garden hose made out of just a layer of rubber, and you use that garden hose a lot, I think what happens is you'll find certain parts of the garden hose end up getting big bulges, or they end up rupturing and they leak. So a lot of garden hoses, like you buy from Home Depot or Lowe's or City Mill, you'll notice that it's rubber, but it has some sort of mesh of wires wrapped around it also. And that provides structural support so that the walls don't collapse. So that's what these pericytes do. So you can see in normal capillaries, you would have them wrapped with the pericytes. The pericytes are kind of wrapped around almost like a wired mesh, holding the cells together. On the right is a typical capillary running through a large tumor. Two things. One, you can see the pericytes are having a hard time hanging on. So it's almost like they're falling off. So they cannot structurally hold the integrity of the capillary wall. and the capillary itself is much bigger. So tumor capillaries typically can get up to 3 times the diameter of normal capillaries. So that means more blood flowing through them, and more blood and other types of nutrients, gases are leaking out of them, including all kinds of growth factors and things like that, that end up in sort of a feedback loop, encouraging even more capillaries